A very good morning to doctor and friends. Today our group will going to present the infographic about enzyme. We are from SPK 1013 Group A. And today we will further explain about enzymes and classifications, unique properties of enzymes, enzymatic reaction and regulations, the importance of enzyme and regulations for living organisms. So, here we go. Classifications of enzyme. The International Union of Biochemistry (IUB) system recognizes that there are only 6 basic reactions catalyzed by enzyme. The first classes of enzyme is enzyme oxidoreductase. Enzyme oxidoreductase catalyze oxidation or reduction reactions. Hydrogen, oxygen, or electrons are transferred from one molecule to another. The first one reaction is oxidase catalyze the addition of oxygen. For example, glucose plus oxygen will produce gluconic acid and water. The second one reaction is dehydrogenase catalyze the removal of hydrogen from the substrate, passing it to a hydrogen acceptor. For example, malate plus NAD plus will produce oxaloacetic and NADH and hydrogen acceptor. The third classes of enzyme is hydrolase enzyme. Hydrolase enzyme catalyze the hydrolysis of various bonds. Hydrolysis occurs by the addition of water. Most of the digestive enzyme, pepsin, lipase, amylase, and also hydrolase. For example, maltase and water will produce glucose and glucose in the presence of catalyst maltase. The fourth classes of enzyme is lylase. Enzyme lylase craft various bonds like carbon to carbon, carbon to nitrogen, carbon to oxygen, and carbon to sulfate by means other than hydrolysis and oxidations. For example, decarboxylase means remove carbon dioxide, and carboxylase fix carbon dioxide means. For example, pyruvate plus coenzyme A plus NAD will produce acetyl coenzyme A and NADH and hydrogen acceptor and carbon dioxide in the presence of catalyst pyruvate decarboxylase. And for another example, pyruvate will produce ethanol and carbon dioxide in the presence of catalyst pyruvate decarboxylase. The fifth classes of enzyme is enzyme isomerase. Enzyme isomerase catalyze isomerations means atoms or groups are rearranged within the same molecule. For example, glucose 1-phosphate after rearranged will become glucose 6-phosphate in the presence of catalyst phosphoglucosamidase. The last one classes of enzyme is enzyme ligase. Enzyme ligase catalyze the reaction that join two molecules with covalent bond. For example, 2 DNA and ATP joined together will become longer DNA plus ATP plus phosphate group in the presence of catalyst DNA ligase. Thank you.
Many enzymes require the presence of small non-protein units. Isoenzymes are different forms of an enzyme. A complete catalytically active enzyme together with its coenzyme metal ion is called a induced fit model states that the binding of substrate induce a conformational. According to Locke and the Key model, the shape of the substrate and the active site of the enzyme.
Hi, I am Su Xiangshen. Today I will be presenting the enzyme reaction and regulations. First of all is the Michaelis-Menten kinetics. Michaelis-Menten kinetics is the measure of the substrate binding affinity. Okay, here shows the graph of Michaelis-Menten kinetics where the reaction rate of enzyme against substrate concentration. The Michaelis-Menten plot is increasing gradually till it reaches Vm, which is maximum velocity. The Vmax is the maximum velocity, which means the maximum rate at which a reaction can occur with the given enzyme concentration. The Vmax of an enzyme reaction doesn't increases no matter how much substrate concentration you add in. Okay, here we can find out the Km from half of the Vmax. Km is the Michaelis-Menten constant. With the Km, it can help us to identify the affinity of substrate enzyme. By the meantime, while the Km increases, the affinity decreases. We'll move along to the enzyme activator and enzyme inhibitor. The definition of enzyme activator and enzyme inhibitor are: enzyme activators are a chemical species that can bind with an enzyme to increase its activity, while enzyme inhibitors are chemical species that can bind with enzyme to decrease its activity. The effects on the enzyme activator can increase the activity of an enzyme, while the effects on the enzyme inhibitor can decrease the activity of an enzyme. So the example of enzyme activator is glucokinase, and the example of enzyme inhibitor is ribonuclease. Here are the three reversible enzyme inhibitor. The first one is the non-competitive inhibition. The non-competitive inhibitor will bind at the inhibitor binding site. While the inhibition occurs, the substrate no longer fits the binding site shape. Binding of the inhibitor causes the substrate binding site to change shape, so the chemical reaction is not carried out. The second is the competitive inhibition. As we can see, both the substrate and the inhibitor have the same shape, causing both of them to compete for the substrate binding site. So when inhibition occurs, if the inhibitor binds to the enzyme, the chemical reaction which would be carried out by the enzyme does not happen. The reaction is not carried out as no substrate can bind because the inhibitor has bind on to the enzyme. And lastly, end product inhibition. End product inhibition occurs when too much of the end product is created, causing there to be an excess. The extra product will bind to the allosteric site, changing the shape of the substrate binding site, so the substrate can no longer bind, stopping the metabolic pathway. And lastly is the allosteric regulation. Allosteric regulation is where the regulatory molecule binds to an enzyme someplace other than the active site. The place where the regulator binds is called the allosteric site. Pretty much all cases of non-competitive inhibitions are forms of the allosteric regulation. So here is an picture of the allosteric inhibition and activation. Okay, that's it from me. Thank you.
Assalamualaikum. My name is Muhammad Firas Binuddin, and here I want to explain about the importance of enzyme and regulation for living organism. First, the importance. Enzymes play an important role in metabolism, diagnosis, and therapeutic. All biochemical reactions are enzyme catalyzed in the living organism. Level of enzyme in blood are of diagnostic importance. Example, it is a good indicator in disease such as myocardial infarction. Enzyme can be used therapeutically, such as digestive enzyme. Next, role of enzyme in living things. Enzyme catalyze all metabolic reactions. They lower the activation energy. The energy input needed to bring about the reaction. Regulate to thousands of different metabolic reactions in a cell and in the organism. The activity of cell is determined by which enzymes are active in the cell at that time. Cell activity is altered by removing specific enzymes and synthesizing new enzymes. Okay, next. Enzyme regulation such as allostatic regulation. Here you can look the picture. That is allostatic regulation. Next, reversible covalent modification. And the last one, proteolytic cleavage. Okay, that's all from me. Thank you.